So let's start with identifying the individuals. In this case, we know that there was a total of 20 healthy mice that were then split into two groups, a group of 10 that received tumor-specific antibodies as a potential treatment for cancer, and another group of 10 that received just some basic treatment that should not be effective. Those two treatments can be considered one variable, a categorical variable, treatment 1, treatment 2. What else is shown here? We recognize those two groups, the two conditions imposed on those mice. And then we see in the vertical axis something about number of mice with lymph node metastases. So a variable is something that varies from individual to individual. Obviously, if you think about just one individual, so in this case, one mouse, we are not going to record something about the number of mice that have metastases. So this here is a summary of a variable, the variable being whether or not any given mouse has lymph node metastases. So our variable presence of metastases is categorical because for any one mouse, it can only be yes or no. To understand all of the values that are cited here, we can see that the out of 10, this is out of the sample size, how many individuals were given any given treatment. And then we have the actual summary, so number of mice that do have metastases. So the summaries here are counts for the outcome, yes. So the main variable, the response variable, is the presence of metastases, categorical. And you can think of the treatments as another variable. In the second part of the figure, in graph B, we still have the two conditions imposed, the two groups. And now we see that in the vertical axis is showing total number of lymph node metastases. So it's those numbers. And the detailed symbols represent the number of secondary lymph nodes or lymph node metastases detected in each mouse. So a variable is whatever we record for each individual. Therefore, this is exactly what was recorded here, the number of lymph node metastases for each mouse. So in addition to treatment, we have another variable here, which is the number of metastases found. And the possible values are 0, 1, 2, ... So this is a discrete quantitative variable. The numbers circled here represent the total for each group, which, as you can see here, for the tumor-specific antibody treatment, almost all of the mice had no metastases, so zeros, but one mouse had some, and it had exactly one metastase. Therefore, the total is 1. You might want to think about how the data would look like if you were to have a data table ready for analysis. So the best way to organize a data table is to have one row for each individual. So here we have a grand total of 20 mice. We know that 10 of them had the control treatment, and the other 10 had the tumor-specific antibody treatment. Two variables were represented. One was the presence of metastases, yes versus no, categorical. The other one was how many metastases, that's a number. We can now interpret the findings of this study in context. So first of all, this is an experiment where the individuals were randomly assigned to those conditions. And what this experiment found was that the mice that received the tumor-specific antibody fared a lot better than the mice who received the control. In the control group, all of the mice had metastased, whereas only a tiny fraction, 10%, had metastased in the tumor-specific group. We can also see that the tumor-specific antibody group, the treatment group, had much better outcome on average, with the mice in that group having only an average of 0.1 metastases per mouse, compared with 2.4 metastases per mouse for the control group. And lastly, we also noticed that the results were much more consistent in the treatment group than in the control group. Notice that the variability is clearly greater in the control group. This is visible in the dot plot. It's also visible in the value of the standard deviations, clearly greater in the control group than in the treatment group, as well as the interquartile range or the whole range